everyone. I am Dr. Monica, working as Associate Professor in NIT Institute of Pharmacy. I, Today I am going to start with my subject that is known as Pharmaceutical Microbiology, having code BP303T. Now, the contents which I am going to cover under pharmaceutical microbiology are: first, microbiology; second, I will like to cover branches of microbiology. After that, I will like to focus on, that is, eyes in microbiology. After that, the role of microbiologists we will cover, and we will try to cover the history of microbiology. Under history of microbiology, we will try to cover the discovery era and the spontaneous generation theory. As we all know what is pharmaceutical microbiology, I will try to let you know about first what is microbiology and how we can apply the concepts of microbiology in pharmaceutical industry. So, in the contents, as I have told, I will cover first microbiology and what is the main definition of microbiology and what microbiology stands for. Second, I will try to cover the branches of microbiology. What are the various branches which are there within the microbiology field? After that, I will try to focus on 4 eyes, that is, the whole microbiology revolve around these 4 eyes. So, we will try to understand the concept of eyes, what eyes stands for in microbiology. After that, I will try to give a focus on the microbiologist, that is the person who are dealing with the microbiology and the scientists who are working in the field of microbiology. So, I will try to give instructions regarding the research scientists who are working under the field of microbiology. After that, I will try to focus on the history of microbiology, what scientists have done. Until and unless people do not know what scientists have done before in this field, we will not be able to apply the concepts further, we will not be able to do research anymore. So, that is why history is the most important part by which only we get to know, we come to know about what scientists have already done in this field, and we will be able to do more researches and we will be able to find out the maximum results. We will be able to solve many queries if we know about the history and how scientists have worked before. After that, under history of microbiology, in this first lecture, I will try to focus on the discovery era and spontaneous generation theory. Discovery era, we will like to focus on that is microscopes. Until and unless we are not able to see those species, until and unless we are not able to see those creatures, we will not be able to understand their structure, we will not be able to understand what's their shape is, what's their structure is, how they are living. And so that's why until and unless we don't have the microscopes, it will be really be difficult to how microorganisms are seen and what impact they are causing to the industry, what impact the microbes are causing to the research. So that's why discovery era is the starting point, where is the era in which microscopes were developed. After that, I will like to focus on the spontaneous generation theory. Before, until and unless we were not able to discover the microscope, the scientists were not able to see these creatures. So, once they were able to see those creatures, then came the spontaneous generation. That's the question came to the scientist's mind, that whether these microorganisms are generating spontaneously or whether they are present, or whether they are generating from the living beings or whether they are generating from the non-living things. So, big question was there, from where these creatures are coming, from where these microorganisms are coming. So, that was what we call as spontaneous generation, in which various scientists worked to find out the reason behind it, from where these creatures are coming, whether they are generating by abiogenesis process or they are generating by biogenesis process, whether they are coming from living beings or whether they are coming from non-living things. So, these all parts we will cover under today's lecture, that is the first lecture related to your pharmaceutical microbiology field. So, first of all, I will like to tell you about what is microbiology. Now, microbiology is the study of microorganism, in which we study microorganisms. The microorganisms are the creatures which can't be seen by the naked eyes, which are very small, which are usually less than 1 millimeter in diameter, which cannot be seen by naked eyes. They require some kind of magnification, they require some kind of microscopes to be seen, and those creatures are generally what? These are categorized as viruses, they are bacteria, they are fungi, they are algae, they are protozoas. So, these all creatures come under the field of microbiology, in which we study about microorganisms, in which we study about these creatures, that is viruses, bacteria, fungi, algae and protozoas. These creatures can't be seen by naked eyes. So, that is why we require some kind of magnification, we require some kind of microscopes to see these living creatures. So, next I would like to focus on the branches of microbiology. Microbiology can be divided into various branches. Now, how we can divide these micro and how we can put those microbiology concepts under various branches. So, under this, microbiology has been categorized into two. One is basic and second one is applied. Now, what we mean by basic? That means pure microbiology. Microbiology is basic or applied. Basic means we will study about the pure microbiology in this branch, and in applied branches we mainly focus on the application part, where we can apply the concepts of microbiology. So, basic microbiology, under basic microbiology it is further categorized into three parts. One is by organisms, second is by process, third is disease related. So, basic and pure microbiology has been further categorized as by organisms, by process or disease related. Similarly, application based microbiology or applied microbiology has also been divided into various categories, like first further categorized into disease related, environmental related or industrial microbiology. So, under branches, if we focus on the basic part, or if we categorize our microbiology in pure form. So, in the pure form there is first bacteriology, second phycology, third mycology, fourth virology, parasitology, protozoology. So, this is a field in which we study about bacteria. Bacteriology stands for bacteria, in which we study the structure, shape, application of bacteria. And what we know, understand about phycology. In phycology, we mainly focus on the creatures which are algae. In mycology, we study about fungi. In virology, we study about viruses. In parasitology, we mainly focus on the parasites, and in protozoology, we mainly focus on the protozoas. So, all these are studied under pure microbiology on the basis of the organisms, on the basis of the creatures. It has been divided into various categories like bacteriology, phycology, mycology and virology, after that parasitology and protozoology. If we go with process, on the basis of process, the microbiology has been categorized into microbial metabolism, microbial genetics. So, we study about the metabolic process within the that is this branch. In this branch mainly what parameters are there for considered under metabolism of microbes. So, metabolics is studied, and under microbial genetics mainly focus is given to the that is genetics part. And under disease, if we go for the disease related microbiology, this is the main branch which mainly deals with our pharmaceutical field. So, we have to focus on these branches like disease related microbiology. In this, first is immunology, second one is epidemiology, and third one is etiology. Now, what is the difference between immunology, what is the difference between that is epidemiology, and what is etiology? So, if we go for diseases, microbes are harmful, microbes are beneficial. As we all know, they can provide, they have benefits too, as well as they are harmful to us all also. So, if we see immunology, in immunology we mainly study about the immune systems, we study about how our body reacts towards such type of microorganisms and how our immune system develops. So, immunology is a branch in which mainly focus on the immunity part. Epidemiology, in epidemiology, as we all know, in today's era pandemic is going on. So, what does that mean, pandemic, and what does that mean, epidemiology? So, epidemic means is related to certain area. Epidemics means a disease which is related, which is found in a particular area, it is not found in the whole world. Whereas pandemic is a condition which we can see that spread at the most advanced stage, and we are facing that problem of COVID in our era. So, that is what we call as pandemic. That disease is that multiplication is going on at a very fast rate, and we are seeing people which are got affected with that virus, that is known as pandemic. And epidemic is just in a particular area that exists. And etiology is a branch in which we mainly focus on the causes, why this disease is being generated, how it is impacting the people, how it is impacting the economy. So, etiology, how it impacts the environment पे कैसे effect हो रहा है. After that, in disease related microbiology, if we see on the basis of application based microbiology, is further being divided into disease related, environment related, industry related. Industrial based application, disease related application, and environment related applications. So, on this basis, this microbiology has further been divided, as in disease related we mainly focus on the infection, what infections are caused due to some of the microorganisms, what type of infections are generating, and what chemical agents, what chemotherapeutic agents, what antibiotics we can generate from these type of and we mainly study those chemotherapeutic agents, how chemotherapeutic agents can be designed, how chemotherapeutic agents or antibiotics can be generated to cure those infections. So, infection control is always studied under application part. That is also we call it as medical microbiology or pharmaceutical microbiology, as we are pharma people. So, we are mainly focusing on the generation, on the research, how we can find, how we can generate those drugs, how we can generate those vaccines against these type of infections. So, we need to cure those infections. So, studies being done on the application part, we must how we can apply this and how we can find it out the solution for any of the disease. And under environment microbiology, the focus is mainly based on that is what we call as environment, how the drugs, how the that is microbes are impacting the environment, how they are impacting our soil, how they are impacting our air, how they are impacting our whole environment being affected by the microorganisms. So, that is mainly studied under environment. Environmental scientists mainly work on these conditions, ki how microbes are impacting those that is environment. After that, there is a branch which is called as industrial microbiology, and in industrial microbiology main focus is always given on the food beverage industry, pharmaceutical microbiology, and genetic engineering. We can apply our microbe microbial concepts to the genetics part. Even in biotech department also, these microbial studies have been applied. We study microbiology subject in biotech, we study microbiology subjects in pharmaceutical microbiology, as well as we study, that is, microbiological concepts in food and beverage industries also, because most of the, if we see, dairy products are there, fermented foods are being prepared. So in all we require microbes, we need to focus on the microbes. So that was what I was telling, that how what branches are there in microbiology and how we can study, and what application part is being studied under various branches. Next, I want to move toward the 4 I's. In the starting slide itself, I told the whole microbiology moves around these 4 I's. Until and unless we don't know about these I's, that what these I's stand for, we won't be able to understand the actual microbiology. So actual microbiology, if we want to study about bacteria, we want to study about that is virus, we want to study about protozoas, but though before we don't know the processes to study, we don't know how we can generate the pure cultures of those species. So we won't be able to understand. So the whole microbiology always revolve around these 4 I's. Now what are these 4 I's? First I is inoculation. Now what we mean by inoculation? Inoculation is the process. It is the process of transfer, in which we just transfer our microorganisms, we transfer our species, we transfer our creatures from one media to the other. Inoculation is just a process of transfer, what we call as, that is process by which microbial culture is being transferred to sterile nutrient media. Nutrient media में transfer किया जाता है using various sterile inoculating loops, using various sterile needles. Inoculating loops are available by which we can transfer our creatures, our species from one media to the other. So inoculation stands for, that is transfer, that mainly deals with the, that is transfer process, in which we just transfer our creatures, our microbes from one media to the other media. After that comes isolation. Now what isolation stands for? In isolation, when we study about isolation, there are 2 terms which are coming. One is pure culture and second one is mixed culture. Now what we mean by pure culture and what we mean by, that is, mixed culture? Pure cultures are the culture which contains single species, which contains single type of microorganisms. These are being developed from the mixed cultures. We always try to develop pure cultures from the mixed cultures. And mixed cultures are the cultures which contains lot of microorganisms. If we see soil sample, if we see water samples, if we see that is air samples, all samples have mixed cultures. All samples have lot of number of microorganisms, number of species are there within those mixed cultures. So what isolation stands for? Isolation mainly stands for that is isolation of pure culture. We have to isolate a single species from those multiple culture samples in which there are a lot of microorganisms. By using the isolation techniques, we can generate pure cultures, we can generate the cultures in which there will be only one species, one microorganism, that is culture will be one organism only will be there. So isolation is a technique. After that comes incubation. Now what incubation means? Incubation means we have to provide the environment, we have to provide the condition in which these type of microorganisms can grow. So environmental conditions, provide proper environmental conditions for microbial growth. By using the various incubators, by using various instruments, we can provide the humidity, we can provide the temperature, we can provide the pH conditions in which the microbes can grow. After that comes identification. Now, what this identification stands for? Identification stands for identifying, means these will tell us how to identify, what equipments we can use to identify the microorganisms, like microscopes, various staining reagents, various biochemical tests can be performed to identify the microbes. So, these are the 4 eyes around which whole microbiology revolves, that is incubation, isolation, incubation and identification. After that comes microbiologists. Microbiologists are the people who are specialized in the field of microbiology. They are the scientists who deals with the organism. Microbiologists are the persons who deal with the microbiology and are the scientists who work with the organisms, who work with the microorganisms that cannot be seen with naked eyes. Example are viruses, bacteria, fungi, algae, protozoa, etc. So, if we move towards the history part, before starting with the history part, we need to know the role of microbiologists. What is the role of microbiologist is, how the scientists are working, how the microbiologists are being working with those that is organisms. So, moving towards history, under history we have to divide whole history into, that is 6 parts. That is mainly, first comes the discovery era, second comes the spontaneous generation theory, third comes the golden age of microbiology, fourth comes the germ theory of disease, after that chemotherapeutic age and microbiology in 20th or 21st century. Discovery era, in this lecture, that is lecture number 1, I will try to give a focus on the discovery era and spontaneous generation theory. What scientists have done under discovery era and what they have done under spontaneous generation theory. As I was telling, under discovery era, mainly the microscopes were generated, mainly the scientists worked to generate, to develop, to assemble the microscopes, to discover microscopes so that they must be able to see the microorganisms. In the spontaneous generation theory, scientists work to find out the reason how these, from where these microorganisms are being generating, whether they are coming from the living things or they are coming from the non-living things. Under discovery era, the first scientist who worked was Robert Hooke. Robert Hooke was the scientist. He was the person who devised the first microscope. He was the person who devised a single lens by which microorganisms can be seen. Robert Hooke stated the life's smallest structure unit is cell. And second scientist who worked under discovery era was Antony van Leeuwenhoek. He is commonly known as father of microbiology, bacteriology, protozoology. He was first to observe living creature. He was the person who discovered a microscope having magnifying power 50 to 30x magnification. He wrote number of papers related to that. He discovered algae, he discovered protozoas, he discovered fungi. Many species were discovered by this scientist, and that is why he is credited as a father of microbiology, bacteriology and protozoology. After that came the spontaneous generation. Scientists were working to find it out whether the microorganisms are coming from living creatures or they are coming from non-living creatures. So, how the scientists work? First scientist was Francisco Redi, who worked to find it out whether the microorganisms are coming from living creatures or non-living. What he did, he took 3 jars, and in 3 jars, first jar he kept uncovered and he placed mutton in this. Mutton was placed and that jar was open. In the second jar he put mutton and he covered with a lid, and in the third jar he put mutton and covered with a wire gauze or covered with a muslin cloth. What he found, what he observed in his research, that that species was having no microorganism generated in this beaker. There were no microorganisms seen in this after long time, and no microorganism even found in this jar which was covered with a gauze. So, by this study he proved that until and unless these flies do not reach the mutton, the microorganisms will not generate. The microorganism can't generate from the non-living thing, that is the mutton will not generate the microorganism will only generate from the until the living creatures. Once the living creatures will touch this mutton, only then the microorganism will generate. If there is no living creature within the that is jar, if there is no living creature is being able to enter this jar, no microorganisms will generate. After that, Johann Needham, he was the scientist who was against this spontaneous generation. Lazzaro Spallanzani was the scientist who was in favor of spontaneous generation, and Louis Pasteur, he was the person who put a colon against that. He put an end to this spontaneous generation. Johann Needham was the scientist who worked, that is if we see Francesco Redi, he was trying to prove that microorganisms are not generated spontaneously. But Johann Needham was the scientist who tried to prove that microorganisms are generating spontaneously. He took a flask, he put a nutrient broth into that, and he covered that, sterilized that for a few seconds, and after kept it for a long time. What he observed, microorganism will generated. He proved that, he tried to prove that microorganisms are generating from the broth. They are not coming in contact with any of the, that is, flies. And Lazzaro Spallanzani was the scientist who told that they are generating, they are not generating spontaneously. How Louis Pasteur put a told that microorganisms are not generating spontaneously. What he tried to design a special type of a flask. As you can see, this flask was swan-necked flask. Swan-necked flask is designed specially for the proving that our microorganisms are not generating spontaneously, they are generating from the living beings. How he proved? He took the flask, put the flask on the flame, sterilized the broth, the whole broth was sterilized. After that, after many years, he saw that all bacteria, dust particles were getting entrapped within this, but oxygen was able to move the nutrient broth, oxygen was able to reach the nutrient broth. So, by this experiment he tried to prove, if until and unless these bacteria will not reach the nutrient broth, microorganisms will not generate for years. But as he tilted the flask, after tilting, those bacteria reached the nutrient broth, and within hours the microorganisms generated. So, by this he tried to prove that microorganisms are generating from the living beings. Until and unless bacteria are not able to reach the broth, those microorganisms will never generate. So, by this I tried to prove that in the discovery era microscopes were developed, and in that is spontaneous generation, mostly the scientists tried to prove that whether the microorganisms are coming from living beings or non-living beings. So, he was able to prove. Louis Pasteur was a scientist who worked in the golden era, and he tried to prove, he gave proper experimentation that microorganisms are not generating spontaneously, they are coming from the living creatures. Until and unless living creature is not being there, no microorganism will generate, will not develop. So, these are the main references which you need to follow for covering the history of microbiology, or if you want to cover pharmaceutical microbiology, these are the main references, that is Prescott, Pelzar, K.R. Aneja, W.B. Hugo. These are the reference book which you can follow for your pharmaceutical microbiology. So, by this I want to over my lecture based on discovery and this scientists who worked in the spontaneous generation. So, this is what I wanted to tell in the first lecture related to the history part, history of microbiology, in which we have mainly focused on the, that is, discovery era, in which microscopes were generated, and second, spontaneous generation, where scientists worked, where their scientists tried to find it out whether the microorganisms are generating from living creatures or they are coming from non-living creatures, and Louis Pasteur was the scientist who was able to prove that that microorganisms always come from the living creatures, they do not come from non-living things.